So now we've learned how to replicate the DNA, how to make copies of it so that we can make more cells. And we've learned how to decode the DNA and make proteins. So we studied how to do it the right way, the way it's supposed to go. But what if a mistake happens? What if we accidentally replicate the DNA wrong? We change a base or two. Well, when something goes wrong, we call that a mutation. Now, if a mutation occurs in one of your body cells, such as in your skin or your liver or your heart, that mutation is special just to you. And maybe nothing will happen, or maybe it could develop into something bad, like a tumor. Now, if a mutation occurs in a sperm or egg, and then that sperm or egg is used to make a baby, well, then that mutation gets passed on to the offspring. And every single cell in that baby has that mutation, and it could cause something like a genetic disease. We'll learn about that more in an upcoming unit. For now, we need to study what type of mutations occur in the DNA. So let's start with some definitions. First, what is a mutation? A mutation is absolutely any change in the DNA sequence, no matter how small the change. If there is a change in the DNA, then it is considered a mutation. So what causes mutations to occur? Well, sometimes it's just by accident. While you're replicating the DNA, you make a boo-boo, and they just put in a wrong base by accident because it happens incredibly fast. Sometimes accidents happen. Or mutations can be caused by mutagens. So here are some more of your word wall words. So what is a mutagen? A mutagen is absolutely anything that increases the chance of a mutation occurring. So there are certain chemicals that are known as mutagens. They can cause cancer. UV light, so laying out in the sun, we all know causes skin cancer. And that's because UV light is a mutagen. Radiation is a mutagen. Okay, so anything that causes mutations to occur more often. So when a mutation occurs, one of three things can happen. A lot of times mutations have little or no effect. You don't even know that a mutation has occurred. And sometimes you can tell the mutation occurred, but it doesn't matter. For example, blue eyes. Having blue eyes is actually a mutation. Humans started out having just brown eyes. A mutation occurred that led to blue eyes. And blue eyes isn't harmful. It doesn't affect you in any way to have blue eyes. It's just different. So it just provides a little variation or diversity. Now, very rarely, a mutation can be beneficial. It can be a good thing. It can make the organism with that mutation survive better. For example, being able to digest dairy. So if you can eat dairy products like milk and cheese and yogurt and ice cream without any problem, no effects, well then you're mutant. Being able to digest dairy is actually a mutation in humans. It's not normal. I mean, it's normal now because it's pervasive, but it originally started as a mutation and it was beneficial. People who could digest dairy had stronger teeth, so they kept them longer. They had stronger bones, so they lived longer. So it was a benefit. It was a good thing. But more often than not, mutations are deleterious. They're bad. They're harmful, such as the genetic diseases that we're going to study soon. Cancer, tumors, those are mutations that are bad. All right, so we need to study the types of mutations. You want to make sure that you know the definition of the mutation and what's happening, how it's different, how the mutated DNA is different from the original DNA. And that's part of what our worksheet's going to be today as well, is practicing looking at an original strand of DNA and then a mutated strand and saying, okay, what happened here? What type of mutation? So the first group of mutations we're going to study are called point mutations or substitutions. Okay, point mutation and substitution are two words that mean the same thing. It means that you have changed one single base for a different one. So you've changed one nucleotide. Okay, remember a nucleotide is a phosphate, a sugar, and one of those 4 nitrogenous bases. So what's happened is you've changed one base. So maybe there was supposed to be an adenine, but for some reason, accidentally, we put in a guanine. Okay, so we switched an A for a G. Okay, one single base change. That is a point mutation. 1 point change. It's also called a substitution because you're substituting one base for another. There are 3 types of point mutations. One is known as a missense. With a missense, when you change that one single base, you are changing the codon. So the code word is now for a different amino acid. Remember our little codex that we used yesterday when we were looking up the 3 letters to see what amino acid we had. So whereas the first time we might have coded for a lysine, whoops, because of this single change, now we're coding for a serine or something like that, okay? So look back at that amino acid code chart so that you can sort of make sure you understand what I'm talking about, okay? So missense, you made a little mistake. Okay, and you change one amino acid for another. Missense is a small mistake. Then you have a nonsense. With a nonsense, what happens is where you should be coding for an amino acid. By changing that single base, you change it from a code word for an amino acid to a code word for stop. So what happens is the ribosome reads stop, and it stops making the protein, which means the protein gets cut short. This is almost always really, really bad, and the protein doesn't work at all. Okay, so nonsense because the protein don't make no sense. Okay, because it got cut short. So a nonsense makes no sense because it got cut short. And then the third one is silent. Remember that the amino acid code is redundant. Okay, there are multiple codons that code for the exact same amino acid. So when you have a silent point mutation, that change that you made, you still code for the exact same amino acid. So the protein is still made correctly and it still works, but the coding is just different now. So it just provides a little diversity, and when a silent mutation happens, it stays in the genetic code, and it could get passed on and lead to more changes down the road. So these are your 3-point mutations. Okay, here's a picture example for you. So you see here, this is the original code with a T paired with an A and a C paired with a G. Now at the bottom, we made a whoopsie, and we put a C here where there should be an A. Now this shouldn't happen because T and C, they don't go together, right? And what happens is these two cannot hydrogen bond together. They don't fit together like puzzle pieces. So you're actually going to have a little bubble in the DNA here, which is usually a good thing because a lot of times we can fix that because there's these little proofreading enzymes that go along and look for mistakes like that, and then they fix it. The problem is, which one do they change? Do they get rid of the T or do they get rid of the C? They have to guess. They got a 50-50 shot to know, and it's by chance whether they get it right or not. Most of the time, your cells do a really good job fixing the DNA and copying it correctly. Here's another example. Here, there's supposed to be a G there. Whoopsies, we put in a C, so this is a missense, because now we're just going to code for a different amino acid. Okay, but see, it still goes on. You just have one amino acid change. But look down here at the nonsense. The mistake we made now codes for stop. The rest of the protein, it doesn't get made. This is nonsense because it don't make no sense because the protein doesn't get finished. An example of a disease that is caused by a point mutation is sickle cell anemia. This is where your red blood cells don't get made correctly because it's the gene for how to make red blood cells. They're supposed to look like this, but they look like this. Okay? These get tangled up and cause a lot of pain in people who have sickle cell anemia. Our next group of mutations are called Frameshift mutations, and there are two types. You can have a deletion or you can have an insertion. Okay? And what happens with the Frameshift mutation is you put in one extra codon, or you take out one, or not a codon, I'm sorry, and a single amino acid. You add a single amino acid, that's an insertion, or you take out a single amino acid, that's a deletion. Now what happens, remember, your ribosome reads the nucleotides in sets of 3. So when you add one extra, or you delete one, you shift the reading frame, and it messes up every single amino acid from that point on. So this is pretty catastrophic as well. Okay, so you can see up here, this is the original strand of DNA. It should be CTGAGT, but we had a whoopsie, and now it's CTGAT. We inserted an extra T, GT. Down here, whoopsies, we were supposed to have the A, we accidentally deleted it. Okay, this is also going to create a bubble in the DNA, which can usually get fixed. And again, 50-50 shot of whether we fix it correctly or not. My favorite way of putting this into mindset of how this messes up everything from then on out is think of a sentence that's only made with words using 3 letters. The fat cat sat. That's how the ribosome reads, in 3 letters. Delete one letter. Still read in sets of 3. Hef, atk, at. That's not a sentence. That makes no sense, right? That's what happens to the protein. The protein is just completely destroyed because it's not made correctly. Okay, same idea. That sentence helps me so much. Our last type of mutation is called a chromosomal inversion. What happens here is an entire chunk of DNA gets knocked out, and the cell trying to fix the DNA accidentally puts it in reverse. So it gets knocked out, flipped around, and put in backwards. That's what inverted means. It means backwards. So chromosomal inversion means that part of the DNA is in backwards. So if you look down here, you can use either the numbers or the colors. If we number the sets, it's 1, 2, 3, 4. Well, if 2 and 3 get knocked out and then chromosomally inverted, they get put back backwards. Now you have 1, 3, 2, 4. Okay? So this is a bad thing too. So chromosomal inversion. A chunk gets knocked out and put back in backwards. Okay, here you can see it marked with letters A, B, C, D, E, F, G, H, I, normal alphabet. Knock out a chunk, put it in backwards. Now we have A, B, C, F, E, D. That part's backwards, G, H, I. Okay, chromosomal inverted. It got inverted. It got put backwards. Okay. So when a mutation occurs, can you fix it? And the answer is yes, sometimes. Most of the time. You get mutations in your cells every single day. But most of the time, your cells fix it. Okay, most of the time it gets fixed and everything's fine because you have special enzymes that after the DNA is replicated, these enzymes zip along the DNA and proofread. They look for those little bubbles that happen if you pair up things wrong, or the little bubbles that happen if you insert or delete. Okay, but they don't always work. And when they don't work, well, and mutations stick around, well, sometimes that can lead to cancer.